Hi, so if you were absent for the notes in class this week, this is part 2 of the cell cycle notes, and today we're going to be talking about regulation and cancer. So let's first talk about what controls cell division in eukaryotic cells. And the answer to that is very simple. The answer to what controls it is proteins. And those proteins are going to regulate the progress of cell division at certain checkpoints. So those checkpoints you can see represented over here in this little diagram on the right by those stoplights. So there's one here, there's one there, and there's one there. The first checkpoint is called the GAP1 or the G1 checkpoint. So on that diagram, which looks a little bit different, but it's the same diagram, that's this checkpoint right here. So that's at the end of the G1 phase, which is the cell growth. Remember, right here is the starting point for a cell's life. So in this phase, that cell is growing, okay? And at the end of that particular phase, the protein are going to check to see if that cell is healthy and has grown appropriately. At that point, it's going to determine if a cell should divide, enter G0 phase, which is normal cell function. So essentially the cell isn't doing anything to prep for division, or if that cell should delay division temporarily. So at this checkpoint, there's 3 options. That cell could either decide at this checkpoint, hey, I'm going to divide. That cell could decide, hey, I'm just going to do nothing and do my normal cell function things, whatever that might be. Or the cell could decide, hey, I'm going to divide, but not yet. I'm going to delay that for a second. Okay. The next checkpoint is the G2 checkpoint, okay? And that checkpoint is all the way over here at the end of the G2 phase. So to give you an idea of where we're at, again, this is start. So the cell has gone all the way through the G1, it's grown. It's gone all the way through the S phase where that DNA replicates, and then through that G2 phase all the way to the end. And at the end, the DNA repair enzymes are going to check to make doubly sure that that DNA replication that was done all over here in the S phase was done correctly. Because if the DNA replication is done wrong, if the cell makes a copy of that DNA incorrectly, that can cause a lot of problems down the line. So what it's doing is it's screening for something called mutations, which are changes in the genetic code. Then our third checkpoint is the mitosis checkpoint, so that's right here. Some proteins are going to signal the cell to continue mitosis. So in the mitosis phase, this phase right here, the cell's already trying to divide. The nucleus is starting to divide. But a little way through mitosis, those proteins need to check to make sure that it's going to divide correctly. So what they're checking to see is that the spindles, those little fibers that attach to the chromosomes, are set up correctly. Because if they're not, then the chromosomes divide unevenly. And again, that can be deadly for cells if the chromosomes do not divide correctly. Okay, so here is a diagram that's a little bit more well put together, in my opinion, because it tells you pretty much everything that's happening. So here's your first here's the checkpoint at the end of mitosis, here's the checkpoint at the end of G1, here's the checkpoint at the end of G2. And it also tells you what's happening in each of the phases, okay? So this would be a diagram that I would spend some time on before any test, quiz, or assessment comes up. I would look at this to make sure that you know what's happening in each phase and what each checkpoint is doing. But sometimes things go wrong. Sometimes cells don't replicate properly, sometimes things happen. And when that happens, usually the result is cancer. So to really talk about cancer and what causes it and everything, we have to talk about proto-oncogenes and oncogenes. Proto-oncogenes are genes that are regulators of cell growth. So they control how a cell is going to grow. They're going to code specifically for the production of those regulatory proteins. Now, those regulatory proteins are the ones that are controlling the checkpoints that we just talked about. They're going to ensure that cell division occurs properly in the right sequence at the right rate, okay? So these proto-oncogenes are making sure that cells are doing things right. Now, another thing about proto-oncogenes, though, is that they are normal genes. Every single person has them. Every animal has them. Every plant has them. But they can become something else. They can become something called oncogenes. And the way that a proto-oncogene becomes an oncogene is through mutation, which I had mentioned earlier. A mutation is a change in the genetic code. So if you have a gene for cellular regulation and then there's a change in it, a lot of times that change is bad because that change could make the gene not function right, et cetera, et cetera. So if that happens, if the gene is no longer functioning right or if it experienced a change, that can turn your normal proto-oncogene into an oncogene, which is something that can result in cancer. So once again, and I am repeating this because I really want you to know what it is, a proto-oncogene that has undergone a mutation is now an oncogene, okay? And an oncogene, because it's not working properly, is going to cause uncontrolled cell division. When cells are dividing and they should not be. That is what cancer is. When cells are dividing and they shouldn't be. You also have these things called tumor suppressor genes. Now, tumor suppressor genes are genes that are specifically to prevent tumor formation. So if we didn't have these, you might grow random tumors all over the place, but we do. So they make sure that cell division is not occurring too often. It's not occurring way more than it should. So that might sound to you pretty much exactly like a proto-oncogene or an oncogene or whatever, because if tumor suppressor genes go wrong, then yes, cancer can result from that. So that sounds pretty similar, but here's some differences. An oncogene is going to result from the activation of a proto-oncogene. There only needs to be one mutation for a proto-oncogene to become an oncogene. And here's some examples of oncogene: P53, BRCA1, RB, APC. I'm not going to go into what all of these are because I would be here for a long time, but those are some examples. Tumor suppressor genes, on the other hand, they result in cancer when those genes are turned off. So they're on all the time. These ones are not on all the time. These ones are on all the time. And when they are turned off, then they cause cancer. These ones need 2 mutations to inactivate that gene, whereas oncogenes only need 1 to turn them on. So examples of tumor suppressors are SRC, RAS, HER2, MYC. Again, there's more than that, but I'm not going to go into what exactly each one of those does. So now we've talked about what causes tumors, what causes cancer, but what even is a tumor? A tumor is an abnormal growth of cells that results from uncontrolled cell division. So what you're looking at here in this image, I'm going to outline it for you. This here is a tumor. You can barely see it on this chest x-ray, but it does look lighter than everything around it because x-rays are going to go through less dense tissue more easily than they go through more dense tissue. So this tumor here is more dense than the tissue around it, so it's popping up on that chest x-ray. Here is a diagram of normal epithelium in your colon, kind of going through some changes, and then at the end, cancer comes from that. So this is called a colon carcinoma. So if you or your parents have friends or whatever that have ever had a colonoscopy or a sigmoidoscopy, that means that they're sticking a probe in your colon or your sigmoid colon, and they are checking for polyps. They're checking for little tiny polyps that could become cancerous later. Okay, so here on the left, this is like before there's any danger of cancer. Then you might have ingested mutagens or carcinogens or something, and some abnormal cells start developing. And then you ingest more carcinogens and get exposed to more things that cause mutations, and then a small adenoma forms. Again, this is kind of one of those polyps that they might just take out. It's not cancerous yet. Then again, you ingest some more things that can cause mutations, and suddenly you have a big adenoma, and then eventually that thing is going to become cancerous and start metastasizing elsewhere. So this here, that's a different color, you can see it's starting to invade other places and go downwards into the vasculature, and that is when a tumor changes from just like kind of existing by itself to becoming a malignant form of cancer. Not all tumors will do that. Some tumors are benign, but those that do run the risk of becoming malignant and spreading will do this eventually. So that brings me to the two types of tumors. We could have a benign tumor, which is non-cancerous. That doesn't mean it's a good thing. That doesn't mean it's not dangerous. But benign tumors tend to kind of stay where they're at. They're not going to spread all over the place. So those cells are going to remain within the mass, and they're not going to threaten your life unless they're too big and they're pressing on other things. Or like if you had a benign tumor in your brain and it shoved your brain tissue everywhere, that could be a problem. But like if you had just like a random small benign tumor in your wrist or something, that's really not going to threaten your life unless it starts growing and impeding like you using your hand and a bunch of other things. Okay, a malignant tumor, on the other hand, is a tumor that exists, but also those cells in the tumor are going to break off, travel elsewhere, and form new tumors. And those cells are going to invade other tissues and destroy things everywhere in your body. That process is called metastasis. When a tumor metastasizes, a part of it's breaking off and spreading somewhere else. Doctors can diagnose that by doing a biopsy, and they'll take a tissue sample of that tumor and they'll look at it and evaluate to see if that tumor is the benign type or if it's a malignant type. So what causes cancer? Once again, to sum up, spontaneous mutations of those genes that code for the regulatory proteins can cause cancer. The mutations can occur as a result of exposure to something called a carcinogen. A carcinogen can be a lot of things. It can be a lot of different things. It's not necessarily only UV radiation, because that's the one that everyone's heard of. Everyone's heard that UV light exposure to the sun can cause skin cancer, but there's a bunch of different carcinogens that you can be exposed to. UV light is one, that's on the radiation scale. If you were to get X-ray after X-ray after X-ray after X-ray, and you had not had that lead vest that they make you wear or protection of any kind, that can eventually cause cancer. Cigarettes have carcinogens in them, all those chemicals that you always read about that are bad for you. Processed foods have some carcinogens in them. That doesn't mean if you eat a hot dog, you're going to die of cancer. That's not what that means. But if you ate nothing but hot dogs for the rest of your life, you might be more likely to get colon cancer from that. But again, just because you eat a hot dog every now and again, that doesn't mean you're going to get cancer from that. I would like to stress that. Cosmetics and cleaning products, if you're overexposed to those things, eventually they can cause mutations that could cause cancer. Viruses can do that. An example of that would be HPV, human papillomavirus. Some of you may have gotten a shot to protect you against that when you were a little bit younger. Certain bacteria can cause cancer later on down the line. But again, just because you've used or have been exposed to these things a couple of times in your life, that doesn't mean you'll get cancer. Some people smoke cigarettes forever and never get cancer from it. They might get emphysema instead. So like, just because you're exposed to these things is not a guarantee of getting cancer. I would like to stress that, because usually when I explain carcinogens, everybody automatically freaks out because they think they're going to get cancer just because they've been around an X-ray before. It's not the case. So radiation, again, UV light from the sun, X-rays, those are radiation carcinogens. This diagram here is an example of what skin cancer can look like. And just because you have an oddly shaped mole on your body, again, that does not make it cancerous. But a doctor, if you wanted to get a mole checked out, they would look for if that mole is asymmetrical, if it had a weird border irregularity, like you see that this border is a different color than that one. They would look for odd color variations, so these two go together a little bit. And they would look for it to have grown from before. So like if the mole started out like this and it's suddenly this size within a year, then they might want to take a biopsy of that mole and check to make sure it's not cancerous. But again, just because you have a weird shaped mole does not mean you have cancer. If you want to get it checked out by your doctor, if you happen to have one of those things, I'm not going to tell you not to, because it's always smart to get things checked, but I don't want to freak you out and make you think you currently have cancer. Chemicals, tar and nicotine, those are carcinogens you find in cigarettes. Asbestos is found in old construction materials. So if you have an older house and you went rooting around in the walls, you might be exposed to asbestos fibers, which can cause cancer eventually. Again, not a guarantee, but it's a thing. Viruses, bacteria, HPV, human papillomavirus. Again, some of you might have gotten the Gardasil shot. I got it when I was in middle school. It's to protect against the human papillomavirus that can cause cancer eventually. Other viruses that can cause cancer, Hep B, Hep C. This is an example of liver cancer here. You see how the tissue over here looks kind of normal, kind of pink, and this looks disgusting. That's an example of liver cancer. Bacteria can cause cancer as well. This comes with a pretty funny story. Helicobacter pylori is a bacteria that can be found in your digestive system, and if you're infected with it, it can cause ulcers. An example of an ulcer would be down here. You see that like gross looking part of the digestive tract. It's supposed to look like this. It's supposed to be smooth. So the guy that was investigating H. pylori he had discovered that it caused ulcers, and to test this theory he decided to do the stupidest thing possible, and he was like, you know what, I'm going to prove that this caused ulcers right now, and I'm going to do that by drinking a sample of Helicobacter pylori. Lo and behold, he got ulcers, and then he took some antibiotics and cured himself of that infection. But that is literally the most irresponsible way to try and prove something ever. But every time I heard that story, it always makes me laugh. So I wanted to share it with you. The last thing I want to talk about is apoptosis, which you may or may not have heard of before. It's programmed cell death. So when a cell becomes damaged or deregulated or whatever, that cell can decide to essentially commit suicide. So let's say that the cell had undergone a mutation and those checkpoints detected that something was wrong. The cell can then decide effectively to off itself, to commit suicide, because in that respect, by doing that, that cell is essentially preventing its host from developing cancer. So like if one cell in your body got damaged, it might signal itself to go through apoptosis so that you don't develop a tumor from that. And when that starts going wrong, then you might develop cancer from that. Okay, but that's not the only reason that apoptosis happens. It can be because of removal of tissues as well. So like this diagram on the left here shows, like on a tadpole, adult frogs don't have tails, and that's because all the cells in that little tadpole tail went through apoptosis and died off. The same thing happens to you in the womb, in the uterus. You have webbed fingers and toes, and you don't have webbed fingers and toes right now, and that's because all those webbing cells went through apoptosis before you were born, and and therefore you were not born with webbed fingers and toes. Sometimes they don't go through apoptosis and that signaling is off. So if there are some children, it's rare, that are born with a little bit of webbing left over, but that's an easy procedure to fix. They don't tend to leave it there because, especially if some of it's already gone away, that tissue can become necrotic and it can cause more harm later. And also it would definitely impede your functionality. Having webbing between your fingers, you wouldn't be able to spread your fingers as wide. That tissue is going to be really fragile, so it's easy to get cut and just infected and all of that type of stuff. So that's going to be it for today. Once again, if you were absent, you should be viewing this video. You should have already gotten the notes with me in class if you weren't, so you don't have to watch this twice. Have a good day.